Ladies and gentlemen, esteemed guests, and dear colleague, we are truly grateful for your presence today. Your time and your interest mean the world to us. At the National Higher School of Nanoscience and Nanotechnology, we believe in the power of innovation and excellence. Here, education is more than acquiring knowledge. It's a journey of discovery, growth, and endless possibilities. Here, curiosity fuels innovation, challenges lead to breakthroughs, and every student is empowered to shape the future. Our commitments extend beyond mathematics and physics and chemistry, but also cultivate essential skills like teamwork, leadership, and lifelong learning. Ladies and gentlemen, it's a great honor to welcome Professor Gianni Ciofani, a leading expert in nanomedicine, biointerface, and space biotechnology. He had some research at the Italian Institute of Technology, focusing on smart nanomaterials for theranostic and space medicine. With the research conducted on the ESS, he has secured multiple ERC grants and published over than 119 papers. He has held a position at top universities worldwide and co-founded Kidaria Bioscience. In recognition of his contribution, he was honored as the Knight of the Order Merit of the Italian Republic. Please join me giving a warm welcome to Professor Gianni Ciofani.
Thank you very much for the introduction. Good morning to everyone.
Good morning.
Good morning. I am going to share my presentation. Can you see in full mode?
Yes, sir.
Okay, perfect. So thanks once more for the invitation. It's a pleasure to be here, even if online with you. And I will try today to give an overview about our activities at the Italian Institute of Technology. So just a few words about our institute. Italian Institute of Technology is a relatively young research center that was established 22 years ago. And our main headquarters is in Genova, in North Italy. And then we have several centers that are spread in all the Italian territory, and they are specialized in different topics. I am the director of one of these centers, and namely the Center for Materials Interfaces, and we are located in Pontedera, that is a small city close to Pisa, the city of the Leaning Tower. Maybe you know it. And the Center for Materials Interfaces, we are studying nanomaterials and their interaction with the living matter. And in particular, I am the principal investigator of the Smart Biointerfaces Research Group. What are we studying in our group? We are essentially a group of nanomedicine, but we are interested in the physical properties of the materials, and in particular to the so-called smart nanomaterials. So nanoparticles that are not simple carrier and simple delivery system of some active ingredient, but nanoparticles that are able to respond to some kind of physical stimulation, modify their functionalities, and giving some cues, some excitation to cells and tissues where they are located. And today I will try to give you an overview and an example for each of these families of smart nanomaterials that we are investigating. So for example, we are active in the field of magnetic nanoparticles. in order to perform magnetic targeting and to perform hyperthermia, because when magnetic nanoparticles are stimulated with the alternating magnetic field, they can develop a heat, and this heat can be exploited, for example, to kill cancer cells. Then, in an analogous way, we are exploiting nanoparticles that are responsive to the light, and in particular the near-infrared region of the light, the near-infrared NIR, because tissues and cells are relatively transparent at these wavelengths, and so we are sure to affect and stimulate just the cells that internalize the nanoparticles. Then we are studying piezoelectric nanoparticles. I think that most of you have heard about piezoelectric materials, that are those materials that are able to convert mechanical energy into electrical energy and vice versa. We are exploiting piezoelectricity at the nanoscale with the piezoelectric nanoparticles, and our aim is to elaborate real nanotransducers, like nanorobots, that are able to electrically stimulate cells when they are mechanically deformed, for example, through ultrasounds, through mechanical vibration, and so on. And then we are active in the field of the so-called nanozymes. What are nanozymes? Nanozymes are artificial enzymes that are able to modulate the oxidative status of the cells of the microenvironment where they are located according to the value of the pH of the surrounding environment. And these nanoparticles are particularly useful in counteract oxidative stress. Oxidative stress is related to an overproduction of reactive oxygen species, and it is a common hallmark of several kinds of pathologies, ranging from simple inflammation to cancer to neurodegenerative disease. And so if we have something that is a very strong antioxidant agent, can be very useful for a wide range of pathological conditions. Our attention, as you will see, is particularly focused on the central nervous system, and how our main projects were and are focused on brain cancer, and in particular glioblastoma multiforme. Glioblastoma is one of the most aggressive human tumor. Fortunately, is not so much common, but it is still a deadly disease. And despite there are some therapeutic approaches, in particular surgery followed by radiotherapy and chemotherapy, the survival after the first diagnosis is extremely low and limited to 1 year, 1 year and a half. So it's really a deadly disease. And this is due to several reasons. First of all, of course, we are talking about the central nervous system in the brain, so it is difficult for the surgeon to eliminate just the cancer mass, leaving unaffected the healthy brain parenchyma without strong side effects. So the surgery is very difficult. Then glioblastoma is characterized by high genetic variability, high topographical diffuse nature, so it is interpenetrated with the healthy parenchyma. And then, of course, because we are beyond the central nervous system, we are to deal also with the blood-brain barrier. The blood-brain barrier is an anatomical and functional barrier that is protecting our brain, but it is also hindering many drugs to pass. So, for example, we can have some drugs that could be effective, but cannot cannot cross this barrier, and so it is impossible to bring high concentration of molecules in the cancer microenvironment. So, our idea was to prepare hybrid magnetic and lipid nanoparticles that would be able to cross the blood-brain barrier, reach cancer cells, and perform a dual therapy. Why the dual therapy? Because they can perform chemotherapy by delivering the drug, but at the same time, thanks to the smart component, thanks to the magnetic nanoparticles, if we irradiate the nanoparticles with the alternating magnetic field, we are able to induce an increment of the temperature. And this increment of the temperature is making cancer cells more sensitive to the chemotherapy drug, and they can help also to directly kill cancer cells through apoptosis. The matrix of these nanoparticles is made of lipid, so biocompatible, biodegradable component, and the lipid component is allowing a high drug payload because usually chemotherapy drugs are hydrophobic drugs. So a lipid nanoparticles is very useful to deliver and protect the drug until it reaches the cancer site. And eventually, the nanoparticles are surface functionalized with a ligand that is allowing the recognition of cancer cells after the nanoparticles cross the blood-brain barrier. This is a bit of characterization of the nanoparticles that we developed. As you can see here, from transmission electron microscope images, we have nanoparticles, the nominal size of which is around 20 nanometers. They are very stable in physiological media with a minus 30 millivolt of zeta potential. And they present a satisfactory surface functionalization, in this case with the AngioPep2. AngioPep2 is a small short peptide that is behaving as a dual targeting agent because it is allowing both the crossing of the blood-brain barrier and the recognition of cancer cells. And then, of course, inside this lipid matrix, we have the magnetic component that is represented by very, very small superparamagnetic iron oxide nanoparticles, the size of which is around 2.5 nanometers. And we are loading also chemotherapy drug. So in this case, nuclein 3A, that is an effective non-genotoxic chemotherapy drug. So by summarizing, we have these lipid nanovectors that are loaded with the chemotherapy drug, with a magnetic component, and that are surface functionalized with this small peptide, AngioPep2, that is allowing the targeting towards cancer cells. What we checked after the physical chemical characterization of the nanoparticles, of course, we performed extensive in vitro experiments, and one of the first experiments was directed to check the selectivity of these nanovectors towards cancer cells. So in order to demonstrate that we have a preferential uptake of the nanovectors by cancer cells, we developed fluidic dynamic experiments by exploiting these in-home made bioreactors. These bioreactors are connected with a microsyringe, and they are allowing to perform experiments in dynamic conditions, so we can simulate the blood circulation through the system. And inside the system, we are allowed to perform multi-cultures. So we cultured healthy neurons, SH, SY, FY. We cultured healthy astrocytes, healthy endothelial cells, and then, of course, human U87 glioma cancer cells. We injected the nanoparticles in the system, and after our experiments, we checked the targeting abilities of these nanoparticles. Here you can see some representative confocal images and the quantitative analysis of these images. As you can see, we have a preferential uptake of the nanoparticles by the cancer cells only when the nanoparticles are surface functionalized with the ligand, with the angiopep2, with the short peptide. So we demonstrated the key role of this targeting agent to allow the preferential internalization of the nanoparticles by cancer cells. As you can see here, we stained in blue the nuclei of the cells, in red the cytoskeleton of the cells, and in green the nanoparticles. The strongest accumulation is occurring here, so by cancer cells when the nanoparticles are functionalized with the targeting agents. And it is very, very clear then from the quantitative analysis. So we demonstrated that our nanoparticles are able to recognize and target cancer cells. But of course, as I said, we have also another obstacle to reach brain cancer, and this obstacle is represented by the blood-brain barrier. As I briefly mentioned, the blood-brain barrier is a functional and anatomical barrier that is formed by several kinds of cells, mainly endothelial cells, astrocytes, but we also have pericytes and microglial cells that are forming a very, very tight and protective layer against external substances that are reaching the central nervous system. And this barrier, unfortunately, is hindering also drugs to reach the diseased site in the brain. So our nanoparticles should behave like Trojan horses and hidden the drug inside their core and transporting this drug across the blood-brain barrier. In order to check the ability of our nanoparticles to cross the blood-brain barrier, we developed this in vitro fluidic model of the barrier that is composed by a static bottom chamber that is representing the brain parenchyma, where we are culturing the cancer cells, the neuron-like cells, the healthy astrocytes, and then an upper chamber that is representing the bloodstream compartment that is connected to a peristaltic pump, and it is allowing the cell culture media to flow and to simulate the natural flow that is occurring in our capillaries. These two chambers are separated by a porous membrane, and on this porous membrane, we are culturing on one side endothelial cells, on the other side astrocytes, and it is representing our in vitro model of the blood-brain barrier. So summarizing, we have two chambers. The bottom chamber is representing the brain parenchyma. The upper chamber is representing the blood compartment. And these two chambers are separated by an in vitro model of the blood-brain barrier. Also, in this case, we are injecting in circulation the nanoparticles, and we are assessing their capabilities to cross the blood-brain barrier model. Once again, here you can see some representative images of the cells that are located in the bottom parenchymal compartment, and in blue again the nuclei, in red the cell cytoskeleton, and in green you can see the nanoparticles. It is very, very clear that the nanoparticles are able to cross the blood-brain barrier model to reach the brain compartment and to maintain their selectivity, because as you can see, we have this strong green signal that is suggesting a strong nanoparticle internalization just in the brain cancer cells that are located after the barrier. And this is, of course, confirmed by the quantitative analysis of the images. And then, of course, we demonstrated the targeting ability, we demonstrated the ability to cross the blood-brain barrier. Now we want to demonstrate the therapeutic ability of these nanoparticles. As I mentioned, these nanoparticles are smart because they have a magnetic component. And this magnetic component is crucial in order to attain a satisfactory therapeutic effect. But how these nanoparticles are working in order to kill cancer cells? So we investigated the uptake pathway of these nanoparticles, and we showed that the nanoparticles, after crossing the blood-brain barrier model and after being uptaken by cancer cells, they are located mainly in lysosomes. Lysosomes, I don't know if you know what are. They are the digestive organelle of the cells. And inside the lysosomes, we have a very, very aggressive, harsh environment because of a lot of digestive enzymes and because of a very low pH values of the microenvironment of the lysosomes. So what is happening? Here you can see a time-lapse video of nanoparticles that are located inside the lysosomes of cancer cells that are undergoing alternating magnetic field stimulation. So in blue, you can see the nuclei of the cells, in green, the nanoparticles, and in red, the lysosomes. We are switching on the alternating magnetic field. We have a local increment of temperature that can be also very high, up to 10 Celsius degrees. And here is what is happening. As you can realize, slowly, the red signal is completely disappearing, and the red signal is corresponding to the lysosomes. So what is happening? The nanoparticles, by increasing the surrounding temperature, are destabilizing the membrane of the lysosomes. The lysosomes are disrupted, and they are releasing their aggressive cargo in the cytoplasm of cancer cells. So all the digestive enzymes are not anymore confined in the lysosomes, but they are in the cytoplasm of cancer cells. As you can see here, where we performed an immunostaining of catepsin B, that is one of these digestive enzymes. And of course, the cell is suffering this situation, is undergoing a sort of autophagy, and this is contributing to the death of the cancer cell. I will skip this data, but as you can see here, this is a simple cell viability testing when we are combining the chemotherapy that is due to the delivery of nuclein 3A with the electromagnetic stimulation. We have an almost complete disruption of the cancer cells. So we are performing, as I said at the beginning, a dual targeting, a dual therapy. First of all, because we are delivering a chemotherapy drug that in principle would not be able to reach the cancer cells. And second, because we are switching on these nanotransducers that are increasing the local temperature, they are disrupting the lysosomes, and they are inducing a sort of autophagy in cancer cells. At molecular level, we also demonstrated that following the double therapeutic approach, we have an overexpression of the p53 marker, that is a marker of apoptosis, and conversely, we have a strong down expression of the Ki67 marker, that is a marker of proliferation. So following this approach, we are able to both slow down the proliferation of cancer cells and to increase their apoptosis, their programmed cellular death. So we demonstrated this approach not only in vitro, but also in vivo on an orthotopic mouse model of human brain cancer. And I will not go into details because of lack of time, but the results were quite impressive because at the end of the 90 days period of experiments, we had a survival rate of the animals that overcome the 75% in the group treated with both nanoparticles and alternating magnetic fields. And as you can see, instead, in all the other control, after 40, 50 days, all the animal diet, in the experimental group treated with hyperthermia and the chemotherapy drug, we had most of the mice surviving at the end of the experiment period. So was a quite good result and a quite good demonstration of the suitability of our approach. Now, I will move to the second class of smart nanoparticles that I would like to introduce you today, that is represented by piezoelectric nanoparticles. As I say, the piezoelectricity is a very well-known, since ancient time, phenomenon that is typical of some materials that are characterized by an asymmetric crystalline configuration to convert mechanical energy into electrical energy and vice versa. So we are exploiting this property at the nanoscale, and we are using piezoelectric nanomaterials that when they are mechanically deformed, thanks to ultrasounds or mechanical vibration, they are forming on their surface an electric field. And this electric field is able to stimulate cells. So we are performing what we are calling wireless electric stimulation, because without needing of wires, without needing of electrodes, we are able to electrically stimulate cells with the combination of piezoelectric nanoparticles and ultrasounds. And of course, this is very useful for all excitable cells, like neurons, like muscle cells, like osteoblasts. But we realized that this approach can be very useful also for cancer therapy. Electric stimulation is quite well known to induce cancer cell stop of proliferation, because electric stimulation is interfering with the cytoskeleton of cancer cells, and the cytoskeleton, of course, is playing a key role during the mitosis, during the cell replication. And so if we are electrically stimulating cancer cells, we are able to slow down their proliferation and to induce apoptosis. So our idea is to exploit piezoelectric nanoparticles and to give an indirect electric stimulation to cancer cells. So we developed these nanovectors that are composed by a polymeric piezoelectric core of PVDF-TRFE, that is a well-known piezoelectric polymer. They are stabilized in physiological condition thanks to a lipid shell of DSP-PEG. They are loaded again with a chemotherapy drug, again nuclein 3A, because our idea is always to perform a dual therapy by exploiting both traditional chemotherapy and physical therapy. And the surface of these nanoparticles is once more functionalized with a small peptide, in this case ApoE, that is allowing the dual targeting, so the crossing of the blood-brain barrier and the reaching of cancer cells, exactly like we just saw for angiopep2 and magnetic nanoparticles. So we have a piezoelectric core nanoparticle loaded with a chemotherapy drug, surface functionalized with a lipid that is linked with a small peptide that is working as a targeting agent. This is a bit of characterization of the nanoparticles, scanning and transmission, electron microscopy, investigation of their stability, and most importantly, evaluation of their crystalline configuration. Because as I said, it is mostly important to evaluate that our material possess an asymmetric crystalline configuration. Otherwise, we don't have piezoelectricity. If the crystal is symmetric, the charge are shielding each other, and we don't have piezoelectric phenomena. So PVDF-TRFE is a piezoelectric polymer that is characterized by several crystalline configuration, but just the beta configuration is that one that is guaranteeing piezoelectric phenomena. So with FTIR and with the Raman spectrometry, we evaluated the presence of the piezoelectric component of PVDF-TRFE, and we demonstrated that our nanoparticles owns this property. And then we started to evaluate the possibility to stimulate cancer cells with the piezoelectric nanoparticles. As I said, our idea is to perform an indirect electric stimulation. So we incubated the cancer cells with the piezoelectric nanoparticles. We applied ultrasounds as a source of mechanical energy to deform the nanoparticles. And through calcium imaging, we evaluated the activity of cancer cells. As you can see, when we are applying ultrasounds without nanoparticles, we have a basic level of calcium ions inside the cells. But when we are applying ultrasounds in the presence of the nanoparticles, we have a strong activity in terms of calcium flows inside the cells. And this is suggestive of an electric stimulation. You can see also here from the quantitative analysis that we have sustained calcium flows following the administration of the nanoparticles in presence of ultrasound stimulation. Then, of course, we analyzed the internalization of the nanoparticles by the cancer cells. As you can see here from these confocal images, we have a strong uptake of these nanoparticles by cancer cells. And it was a very interesting result that we are able also to follow the internalization pathway of the nanoparticles with the Raman imaging, because these nanoparticles are characterized by a strong Raman signal. We have a characteristic Raman fingerprint. So we were able to perform a label-free investigation of the internalization of the nanoparticles by cancer cells. And then, of course, we continued our experiment by evaluating the therapeutic efficiency. I will not go into the details of the obtained data. Interested people can refer to the reference that I'm putting here in the slides. But summarizing the results, we observed that we have a synergistic effect of the delivered chemotherapy drug and of the piezoelectric stimulation that are inducing together a high apoptotic rate, and they are slowing down the proliferation of cancer cells. And these effects were particularly observable at the level of the cytoskeleton, because, as I said, the electric stimuli are in some way interfering with the cytoskeleton conformation in cancer cells. And as you can see here, following the stimulation with the piezoelectric nanoparticles, we have alteration of the polymerization of the actin, and we have a higher concentration of globular actin and a lower concentration of fibrillar actin that is essential for the cytoskeleton formation. And this was happening just when we were stimulating the piezoelectric nanoparticles with the ultrasounds. As a result, this effect has consequences on the migration rate and on the cell invasivity of glioblastoma cells. And the migration and invasion of cancer cells are two phenomena that are strictly related to metastasis. So in this way, the nanoparticles, the piezoelectric stimulation is in principle able also to slow down metastatic phenomena. But the key agent here is the role of the cytoskeleton. With this electric stimulation, we are able to interfere, to perform a perturbation of the cytoskeleton conformation, and as a result, we have these effects. Going a little bit more in depth concerning biomolecular analysis, we perform also extensive proteomic analysis and followed by gene ontology. And really, without going into details, because it would be a really complex and time-consuming, but what we realized that following the piezoelectric stimulation, we have a downregulation of all those pathways that are involved in cell division, so in cell proliferation, in mitochondrial organization, so in the metabolic activity of the cells, and again on the mitochondrial respiratory chain. Conversely, we have an upregulation of all those pathways that are related to autophagy, to response to drug, so a more effective chemotherapy drug, and to cell adhesion, so a less propensity of the cells to migrate and to produce metastases. So the gene ontology following the proteomics demonstrated, gave an explanation to all the experimental data that we collected in the previous experiments. We decided also to check the possibility to interfere with the other component of the brain cancer microenvironment. And so, for example, with the endothelial cells, because endothelial cells are forming new blood vessels that are bringing nutrients to cancer cells. You know, cancer cells are proliferating very, very quickly, very actively, and so they need gas exchange, they need nutrients, and so we have the so-called neo-angiogenesis, so the formation of new blood vessels that are nurturing, that are feeding the cancer mass. So the possibility also to alterate the conformation of these new vessels would be really important. So we decided to apply piezoelectric stimulation also on endothelial cells, typical of the brain cancer microenvironment. And what we realized that indeed piezoelectricity is able to hinder the formation and the proliferation of these microvessels by also interfering with the production of cytokines that are related to the angiogenesis. Here you can see some quantitative data, and here you can see some 3D rendering of the piezoelectric nanoparticles that are interacting and associated to the microvessels formed by the endothelial cells. So we have a strong recognition of nanoparticles by the endothelial cells, and we have an hindering of the neoangiogenesis and of the formation of new blood vessels. As we also saw for cancer cells, also in the case of endothelial cells typical of the brain cancer microenvironment, we have a slowdown of the migration rate and of the invasivity of the endothelial cells. And this was demonstrated also on a 3D spheroid invasion assay, where we saw as the endothelial cells are less prone to form ramification and microtubes following the administration of the nanoparticles and the stimulation with the ultrasounds. So this piezoelectric stimulation is hindering and slow down the formation of new blood vessels that would feed the cancer mass. And this is another interesting side results concerning piezoelectric stimulation. Then I would like to move to other kind of smart nanoparticles, and in particular to some particular experiments that we performed in space. Why in space? Because space is a very harsh environment. So we have been evolved to live on the Earth and not in space. So in space, the body, the organism is suffering an altered gravity condition, so microgravity, but it is also suffering the presence of cosmic radiation. And cosmic radiation are very, very aggressive and cannot be completely shielded by the walls of space vessels. So this combination of microgravity and space radiation is inducing a lot of damages on all the human body. And this has a result, the overproduction of reactive oxygen species. So the astronauts are continuously experiencing a situation of oxidative stress. So the overproduction of free radicals, the overproduction of reactive oxygen species that are very, very dangerous because they are inducing damages to the life macro biomolecules. So they are inducing damages to proteins, to nucleic acid, to lipids. And oxidative stress is also a common hallmark of several pathologies, as I mentioned briefly at the beginning, that are ranging from simple inflammation to cancer to neurodegenerative disease. So if we can find a good solution to counteract oxidative stress in astronauts, hopefully we can also find some solution for typical terrestrial pathology. So this research has a dual scope, dual objectives to both address issues experienced by astronauts in space, but also to address some issues related to oxidative stress in terrestrial pathologies. So this is one of the first experiments, the really first experiment that we performed on board of the International Space Station in 2017. And what we tested was the oxidative stress experienced by muscle cells in space following the permanence on board of the International Space Station, and the possibility to counteract this oxidative stress with smart nanoparticles, with the nanozymes that I mentioned you at the beginning. And this is a short video of the launch of our experiments from the Kennedy Space Center in Florida. So our cells are here on the top of the rocket that, of course, is delivering a lot of other experiments. We assisted to the launch. We prepared the cell cultures. This is the moment that our cell culture started their travel to space. And once aboard of an International Space Station, they underwent 1 week experiments, and they were brought back at Earth. And here we performed extensive transcriptomic analysis to detect the effects of the space, the effect of microgravity and cosmic radiation, and the possibility to counteract these negative effects with our nanozymes. In this case, in this experiment, we exploited cerium oxide nanoparticles. You can see a representative transmission electron microscope image here in this inlet. And cerium oxide nanoparticles are a very, very powerful nanozyme with strong antioxidant ability. And in particular, they are mimicking two key enzymes that are present inside our cells that are superoxide dismutase and catalase. And they are scavenging, they are cleaning the cells from the reactive oxygen species, and they are bringing back the oxidative stress level at physiological condition. And again, without going into details, this is what we demonstrated following these experiments. This is another experiment that we performed a couple of years later, still on muscle cells and brought in space on the International Space Station and treated with the cerium oxide nanoparticles. And also in this case, we confirmed the effects that we saw during the first experimental campaign. So space is triggering the overproduction of reactive oxygen species. It is switching on genes that are related to oxidative stress. But when we are providing cerium oxide nanoparticles, so when we are providing nanozymes, the gene expression level is nearly coming back to physiological condition. Oxidative stress, as I said, is not only involved in muscle degeneration, but is involved also in neurodegenerative disease like Parkinson's disease, like Alzheimer, and many other pathologies of the central nervous system. So our idea in the last two experimental campaigns performed on board of the International Space Station was to test the effect of space permanence on neuronal cells, and then testing the possibility to protect neuronal cells with other kind of antioxidant and nanoparticles. In this case, we switched from cerium oxide nanoparticles to polydopamine nanoparticles. Why? Because cerium oxide nanoparticles are very effective, are very powerful, but they are ceramic, they are inorganic, they are not biodegradable. So if we think to long-term clinical translation, human application, we could have some trouble in proposing cerium oxide nanoparticles. This is why we decided to move to polydopamine nanoparticles. Polydopamine is a safe molecule, is the polymer of the amino acid dopamine. It's also a neurotransmitter. So they are safe, they are highly biocompatible, they are biodegradable, but at the same time, they present very, very strong antioxidant properties. And this is why we proposed them for a couple of experiments on neuronal cells that, again, were carried out on board of the International Space Station. But I would like to spend the last minute of my presentation a bit on this second class of nanozymes that I mentioned, on polydopamine nanoparticles. As I said, they are really, really interesting and smart nanomaterials. First of all, because of their antioxidant nature. So they are able to scavenge reactive oxygen species at high extents, but at the same time, they are biocompatible, they are biodegradable, and they are fully organic. They are very, very easy to be synthesized with the standard Stoeber reaction. And as you can see, you can obtain a high yield production with very uniform size and strong antioxidant activity. And we assessed the antioxidant protective effect of polydopamine nanoparticles, for example, in neuronal cells, if we are giving pro-oxidant insult to the cultures and we are protecting them with polydopamine nanoparticles. As you can see from flow cytometry analysis, indeed, the level of reactive oxygen species are coming back to physiological condition. And at the same time, we are protecting the neural cells, but we are also promoting their differentiation with this antioxidant environment. As you can see here from the immunostaining of the neurites of this model of neuronal cells that are statistically significantly longer when we are administering polydopamine nanoparticles with respect to the control. And very, very interesting, these nanoparticles are able to protect mitochondria of neuronal cells. Mitochondria are the powerhouses of our cells. They are the main producer of reactive oxygen species, but they are also the more sensitive organelles towards reactive oxygen species. And as you can see, when we are administering a pro-oxidant insult, in this case TBH, that is a very, very aggressive chemical agent, the shape of the mitochondria and the membrane potential of the mitochondria is deeply altered. But when we are giving to the cells polydopamine nanoparticles, even in the presence of TBH, of this pro-oxidant insult, we are able to revert the condition of mitochondria to the healthy physiological situation. So polydopamine nanoparticles are very, very efficient, and not only to protect the cell on the whole, but they are also efficient to protect mitochondria, and this is very, very important. But then these nanoparticles present another interesting and smart property. They are able to convert near infrared radiation into an increment of temperature, into heat. So they are absorbing the light around 800, 850 nanometers, and they are inducing a local increment of temperature. And this increment of temperature can be very, very finely tuned by changing the power of the laser that we are using to illuminate the nanoparticles. We were able to assess this increment of temperature intracellularly by exploiting fluorescence temperature sensitive dyes. And we checked the increment of temperature inside the cells that can reach up to 10 Celsius degrees following the stimulation with laser. As you can see, each step of this plot is representing an increment of the power of the laser. So we are really able to finely tune the intracellular temperature by changing the power of the laser that we are using for the stimulation. And this is useful, for example, to stimulate neuronal cells. If we maintain the increment of the temperature within a physiological condition, we are able to stimulate and to trigger neuronal activity with polydopamine nanoparticles illuminated with near-infrared light. And we demonstrated this possibility of triggering cellular activity both on neuronal cells, on muscle cells, and also in vivo on Drosophila insects. And here I'm not showing the data because they are still in work in progress about that. But these nanoparticles are really, really smart because once again, they are completely biocompatible, they are polymeric biodegradable nanoparticles, and they can behave as both antioxidant agents and as nanotransducers to convert light into heat. So in my opinion, this last class of nanomaterials is indeed the most promising, and that we hope to bring soon, at least at preclinical level. So with this example, I would like to conclude my presentation and to leave some space for questions, if you have some questions, some curiosity. And of course, I would like to thank all my group here in Pontedera and all our founding agencies. So if you have any question, any curiosity, I would be really happy to reply.
Inshallah.
Thank you very much.
Thank you so much, Professor. Please wait. Thank you so much, Professor.
Thank you.
Okay. Um, let's turn curiosity into conversation. You know, science is about asking questions. So, what's yours for Professor Ciofani?
Sorry, I didn't hear you.
Okay. Okay. Let's let's turn curiosity into conversation. You know, science is about asking questions. So, what's yours for Professor Ciofani? Any questions, please?
Anything, any curiosity you may have, or if you didn't understand something, anything, tell me.
Uh, first, first of all, I'd like to thank you, and I think I speak for all of us when I say we're lucky to have you here.
Oh,
Well,
thank you very much. Very kind.
thanks. Well, my question is, what are the ethical, regulatory, and affordability challenges in making these technologies accessible for people across different countries? Thank you.
Yeah, this is a strong barrier between experiments in the lab and clinical application. So, first of all, what we are trying to do is to move towards materials that are already FDA or EMA approved. So, towards materials that are already approved by the main international regulatory agencies. So this is the first step. As I showed you, we moved, for example, from inorganic nanozymes towards organic nanozymes. We moved from PDDF, TRFE polymer, and now we are working with biodegradable polymer and so on. So this is the first step. And the second step is trying to perform in vivo experiments and trying also to find some alternative platform with respect to in vivo experiments. I didn't show you today, but a big part of my group is working on organ chips and on microphysiological systems where we are using patient-derived cells in a complex in vitro environment. And we are trying to propose this platform in place of ethically limited in vivo experiments. So these are the first steps and the first barrier that we are trying to handle. So summarizing, by using already approved materials, and also to use patient-derived models that are bringing us closer to the clinics.
Thank you so much for your answer and valuable informations.
Buongiorno signore,
Buongiorno.
voglio essere un po' diversa. Parla una domanda in italiano.
Prego.
Secondo lei, quanto siamo vicini a vedere l'uso diffuso del nanomateriale intelligente nella pratica clinica? Grazie mille.
Prego, rispondo in italiano o in inglese?
So,
English, better.
I will repeat my question in English.
Yes, better. Thanks for everyone.
Okay.
So,
In your
I
opinion,
think.
how
Okay, please.
in your opinion, how close are we to seeing the widespread use of smart nanomaterials in clinical practices? So I'll prefer it in English. Your reply.
Yes, please. I think that we are not aware yet, but smart nanomaterials are already in the clinical practice. Think, for example, to the contrast agents for MRI. They are magnetic nanoparticles. They are super paramagnetic iron oxide nanoparticles that are each day used for diagnostic purposes. So we have spions, we have gadolinium chelated nanoparticles, we have magnetic nanoparticles that are used for hyperthermia, for cancer ablation. So we already have a lot of examples in the clinical practice, but maybe we are not completely aware of this. Of course, all the examples that I showed you today are still a bit far to the clinical practice. Maybe in 5, 10 years, to be optimistic, we can find some of them in the daily use. But nanomedicine and smart nanomaterials slowly are already coming in the hospitals and in the clinics.
Thank you, sir.
Hello, sir.
Hello,
Um,
good morning.
First of all, I want to thank you for your insightful and exciting presentation. It was very exciting
Thank
for
you.
me. Now, as a student in the National Higher School of Nanoscience and Nanotechnology, I am interested in if you can explain the processes that were used to make these nanoparticles, if possible.
Yes, of course. It depends on the nanoparticles that I showed you. So polydopamine nanoparticles are very simple. As I said, they are based on the so-called Sturber reaction. So we are mixing the monomer, dopamine, in ammonium solution. And through reduction of the monomer, we are realizing this polymeric chain that are self-assembling in nanoparticles. So with an overnight reaction, by simply mixing ammonium solution and dopamine monomer, we can obtain the nanoparticles. We wash, we purify, and very easy. Then polymeric piezoelectric nanoparticles are quite more difficult to be synthesized, and generally the work is based on a hot homogenization approach. We are mixing the polymer dissolved in acetone, and we are pouring in a hot water-based solution, and we are performing an homogenization, ultrasonication, and at the end of the process we can purify the nanoparticles. But it is quite difficult, and we have to be very careful in all the parameters that we are using. So polymer concentration, ratio of the solvent, temperature of the reaction, and so on. And eventually the lipid nanoparticles are relatively simple. It is oil in water emulsion. What we are doing is to bring the lipid mixer in at the high temperature in order to have then liquid. Then we are mixing these droplets of oil inside a water solution, and then we are very quickly decreasing the temperature in order to solidify these lipid fat nanodroplets inside the water solution, and the nanoparticles are formed. We can proceed with their purification. And the drug, being hydrophobic, is mixed with the liquid lipid at high temperature, but they are quite easy to be realized.
Thank you, sir.
You're welcome. In any case, I can send my presentation, and there are all the references to all the work that I showed you today, because everything is published. And if you are interested in some specific material and methods or anything else, you can read there, and you can find also my email address. And if you have any doubt, you can write me.
Which one? This. Leader. Sorry, can you hear me?
Yes, perfectly.
Good morning. I must say I'm really excited I was excited, and thank you so much for your fascinating explanation and presentation. I actually have a question considering the side effects of this technology. What are the side effects? Can you hear me?
Yes, side effects. Yes.
Yeah, the side effects of this technology, and how can you, or what is going to happen for the remaining nanomaterials after the operation?
And
Thank you.
this is a very, very good question. You're really targeted the correct point. So, which is the side effects? The side effects are the effects of each kind of pharmaceutical therapy. So we are trying to avoid such effects by targeting the nanoparticles just where they are needed. This is, for example, the role of the small peptides that I showed you on the surface of lipid nanoparticles. We are trying to deliver the effects just to the cancer cells in order to avoid any side effects on healthy cells. Of course, if with the traditional chemotherapy, the drug is not specific and is affecting each healthy cells of the human body. With the nanotechnology, we are trying to limit the effects just towards cancer cells. And concerning what is happening to these nanoparticles, this is one of the other points that pushed us to move towards biodegradable materials. Because if the nanoparticles are not organic, they are not biodegradable, they can give a problem of long-term persistence inside our body. So this is why, for example, we moved from serum oxide nanoparticles to polydopamine nanoparticles. With the examples that I showed you today, all the nanoparticles are naturally excreted or destroyed by our body within some weeks, some months. So they are not giving any concern in terms of long-term accumulation. But of course, this is something really, really important that we have considered for clinical application.
Thank you so much for your time.
Welcome.
Good morning, sir. I want to thank you about the amazing information you gave us and the great explanation. As far as I understood, when you talked first about the blood-brain barrier that prevented the nanoparticles from getting in and attacking the brain tumor, what I really didn't understand is how can these smart nanoparticles really know the place of the brain tumor, because we know that the central nervous system is really complicated. And my second related question is, how long does this operation take? Thank you.
Okay, the first question, I can reply you that the targeting is guaranteed by the surface functionalization. We are putting on the surface of the nanoparticles some small peptides like AngioPep2 or like ApoE, and these short peptides are recognizing some cellular receptors that are overexpressed by cancer cells. So internalization is occurring in all the cells, but it is occurring at a higher extent in those cells that are overexpressing receptors that are recognizing angiopep2 and ApoE. So cancer cells are known to overexpress these receptors, and so we are expecting a higher uptake by cancer cells with respect to other healthy cells. Concerning the duration of the procedure, so in the in vivo experiments that we performed on animals, the duration is quite limited because it is an injection. If we are performing a stereotactic injection, can take up to 20 minutes, otherwise even less.
Okay, thank you so much.
La ringrazio molto, Professor Ciofani. Before we conclude, I want to express my gratitude to all of you for your time and engagement. Special thanks to Professor Gianni Ciofani for sharing his experience and inspiring us with his groundbreaking work. May today's insights encourage us to keep learning, innovating, and pushing the boundaries of knowledge. Thank you so much and have a wonderful day.
Thank you to everyone. Have a good day. Thanks a lot. Bye.